In today's video, we're going to talk about DNA replication. Basically, the purpose for DNA replication is the fact that cells have to make more cells. If you get a cut, you're going to lose some red blood cells from your blood. So somehow we're going to have a cell make more cells. And remember, each cell has DNA on the inside. So these two cells also need the exact same copies of the exact same DNA. So how does that happen? DNA replication. So just a quick overview. If we have two DNA strands with our nitrogen bases in the middle, remember the nitrogen bases are connected by hydrogen bonds. The first thing that happens is the hydrogen bonds have to be broken. When the hydrogen bonds break, what happens is that causes the two DNA strands to separate. So we'll have one DNA strand here and another DNA strand here. So then we have to have a different enzyme come in and make the exact same copies or matches of this DNA strand to match up with this one, and also have another enzyme to match up with this DNA strand. So what we're left with is a cell and another cell, two cells with the exact same DNA sequences. So we have copies of the same DNA in two different cells. Here's a quick visual of what DNA replication looks like. The first thing I'd like to point out is please notice that the enzymes don't work at the end of the DNA molecule and work in one direction. They don't do that at all. What happens is the enzyme will start at random places in the middle of the DNA molecule and work in opposite directions. So right here, even if we had three different spots where we started replication, we would have one enzyme working in this direction, another in that direction, one here, one there, one here, one there. So that's a total of at least 6 different enzymes, and that's a pretty small picture. And they keep working, and they would keep going on through DNA replication until in the end we would have two exact same copies of the DNA molecule, exactly what we're looking for. Here's that same picture underneath the microscope where we have an enzyme that's working right here pulling apart the DNA, and here is that opposite end, that enzyme working in that direction there. How this all works is, remember, we have our DNA molecule with hydrogen bonds between them. The first thing that has to happen is we have to break the hydrogen bonds. Helicase is the name of the enzyme that's going to come through and break those hydrogen bonds. When the hydrogen bonds are broken, the two DNA strands are now separated. They go away from each other. The next enzyme that is going to work here is DNA polymerase. DNA polymerase brings in free-floating DNA nucleotides from the nucleus and will match up those nucleotides to make a new DNA strand using the original strand as a template or as instructions. And of course, all of this happens in the nucleus. So like I said before, we just spoke about helicase being the enzyme that breaks the hydrogen bonds. DNA polymerase matches free DNA nucleotides to match up with the parent or the template strand. But the one that I didn't talk about was ligase. So if we just look at one strand here, what's going to happen is sometimes there's going to be little spaces that happen where these nucleotides aren't connected together. So ligase comes in and is going to bond those nucleotides together and eliminate those spaces between. The replication of DNA is a very, very special process, and the structure of DNA allows the exact copying of DNA to happen. Specifically, the base pairing rules allow the exact copying of DNA to happen. Remember, by base pairing rules, we mean A's always match up with T's and G's always match up with C's. So if we have an A on one strand, we know that a T has to match up with it on the other side. If a G is on one strand, we know a C has to match up with it on the other side. The other thing is the new DNA molecule is half of the parent template and half of the new DNA. So if we draw your attention down here at the bottom, here's our original DNA molecule, A's with T's, C's, and G's. Helicase will come in and break the hydrogen bonds and separate the two parent or template strands. Then DNA polymerase will come in and match up free-floating nucleotides to each parent DNA strand. So here we have two exact copies of the same DNA molecule. See, on this side it's ACT, TGA for this molecule. On this DNA molecule here, we have A's and T's, ACT, TGA. Everything's matched up the way that it needs to be. So we have two identical copies of the same DNA molecule. This process is called a semi-conservative DNA replication process. The reason that we say semi-conservative, semi means half, and conservative means that we're keeping whatever it was. So again, if we look at the final product, so we have two exact copies of the DNA molecule. This strand right here is the exact same strand here of the original. This strand here is the exact same strand from the original. And then we have our two new strands that match up with it. So in each of our two brand new DNA molecules, half is old, half is new. It's a semi-conservative process. So here we have a DNA strand, and we are assuming here that helicase has already worked. So the two DNA strands have separated. There'd be another DNA strand here in the drawing. But let's just look at this one because the same thing is going to happen in both strands. The next enzyme that's going to work is DNA polymerase. DNA polymerase is going to grab a free-floating nucleotide from the nucleus that matches up to each base. So here we need a nucleotide with a G. DNA polymerase does that. After that one, DNA polymerase is going to grab a new nucleotide to match up here, which should have an A with it. DNA polymerase will move up and grab a new nucleotide with a C on it this time to match up with the G, and then match up. If we have an A here, we need a T to match up there. So on and so forth until all of these new bases are matched up. Unfortunately, through this process, you're going at a very, very, very fast pace, which means that you could make some mistakes from time to time. So we have to have a way to deal with these mistakes. The easiest way to be able to deal with these mistakes is using our enzymes. We have different enzymes to proofread and fix any of those typos or mistakes that happen when we copy DNA. We can repair mismatch bases, so if A's and T's are always supposed together, if you have an A and a C, well that really wouldn't work, so we'd have to fix that. Or if there's any abnormal bases. Like here's an example of an abnormal base. There's a bond between two nitrogen bases on the same DNA strand. Here that's called a thymine dimer. So we need to get rid of it. So we have enzymes that are going to cut out on either side of that thymine dimer. And then our enzyme DNA polymerase is going to bring in free-floating nucleotides to match up everything and fill in that space on that DNA strand. And remember that we said ligase comes through and helps bond two things? Well, this is another case where we're going to have a space there and a space there. Ligase is going to come in and fill in those remaining spaces. All of this enzyme action at the end for editing and proofreading allows us to go from a potential 1 in 10,000 bases to have a mistake to go from 1 in 100 million bases before there's a mistake. That's a lot of work to do to make sure that we all have the exact same copies of the DNA in every single one of our cells. And here's just another picture of what it actually looks like. On the left-hand side, we have DNA replication happening under a microscope. And on this side is a cartoon drawing of the exact same molecule. So here in the white shaded portion, that would match up to right here, this space. So here we have helicase pulling apart DNA polymerase, bringing in free nucleotides. Same thing with right here, that would match up with right here. Helicase working, DNA polymerase bringing in free nucleotides. This section here matches up with this section here matches up with this section with helicase here and helicase there to there and there. And right here matches up with this spot. Helicase working, DNA polymerase bringing in free nucleotides. And that is all she wrote.
It's like this and like that and like this and it's like that and like this and like that and it's like this. So just chill till the next episode.